Just say, hey, you're listening to Cult of Reason. Just say something like that.
I can't do it right now.
I believe virtually everything I read, and I think that is what makes me more of a selective human than someone who doesn't believe anything.
Cult of Reason.
We're called monkeys.
Apes don't read philosophy.
Yes, they do, Otto. They just don't understand it.
Cult of Reason.
The only true wisdom consists in knowing that you know nothing. That's us, dude.
I anticipate a deeply religious experience.
Interesting way to open up a conversation.
Hey everybody, welcome to Cult of Reason. This one is about the human gut microbiome. I did this one in a bar, which was more casual and fun for me, but there's a lot more background noise because of that, so I hope that's not too annoying to listen to, but I kind of got used to it after a couple minutes. Just a little bit of background. The microbiome refers to a collection of about 100 trillion microorganisms, like bacteria, viruses, and fungi that we all have living in our digestive tract, mostly our colon. And we're just starting to figure out how this complex ecosystem of mostly bacteria interacts with the human host and to what extent this impacts health and disease. We know that a healthy microbiome is important locally in the colon because if you disrupt it by taking antibiotics, it dramatically increases the risk of an infection with something called Clostridium difficile, or C. diff, which we talk a little bit about in the podcast. This is also true in other organ systems and is why UTIs are common in women that take antibiotics, since the normal vaginal flora is similarly disrupted. What's really interesting, though, is the possibility that these complex interactions with our microbiome may have downstream effects beyond the colon and influence things like autoimmune diseases, metabolic disorders and obesity, and even neurologic and psychiatric conditions. One way this could occur is through the absorption of metabolic byproducts of the bacteria. Another possible mechanism may involve interactions of the microbiome with our immune system, since about 60 to 70% of our entire immune system is located in the lining of our guts. So to talk more about this, I reached out to Tom Schmidt, who is a professor at the University of Michigan in the Department of Internal Medicine, as well as the Department of Ecology and Evolutionary Biology. He is a part of the Michigan Microbiome Project, and his lab is studying how diet, particularly fiber, affects the composition of bacteria and or their metabolic byproducts in the human gut, and how this could be important for something called graft versus host disease, which is a condition that sometimes occurs in patients after a bone marrow transplant. Tom is a very affable guy, and I had a great time chatting with him. It's obvious that he's passionate about his work and really excited about the potential to where it could lead. So without any further delay, here's my conversation with Dr. Tom Schmidt about the human gut microbiome. Cheers.
Hey.
Thank you so much for doing this.
Yeah.
So I was excited to talk to you because I think the three most interesting things in kind of the biomedical realm are stem cells, CRISPR, and the microbiome. And obviously I'm in the field of GI, so I was familiar with the microbiome through all the stuff with C. diff, which we can talk about in a little bit. But what's your research background, and how did you get in the microbiome?
Yeah. By the way, I've got to agree with you that the microbiome, the impact is potentially huge, but we don't know yet, right? And so it's an exciting time to be doing the work.
It's got to be extremely exciting, but very daunting. And I couldn't even, I knew it was complicated. But when I knew I was going to talk to you, I started thinking about it some more. And I'm going to lay out, some of all the issues that make it so complicated in a minute. And I want you to tell me if I'm understating it or maybe overstating it. I don't know.
Right.
But first I want to hear a little bit about your background and how you got into all this.
Yeah. So my background is I started in biology. In fact, I was here at the University of Michigan, I liked science. I liked the experimental part of it. I liked the idea of coming to know the world by poking it and prodding it and seeing how it responds, and that appealed to me. So just sort of went from there, went into graduate school. I had not taken a single microbiology course or a biochemistry course, and so I remember the advisor looking at me and said, "Hey, I don't know how you ended up here, but you've got to take some basic microbiology, some basic biochemistry." And I mean, a light went on for me then, because before that I was studying, or the coursework was animal behavior or plant biology. Things didn't connect for me. But when I got to the level of a cell, like I understood, things made sense then.
Really?
Because like if you knew how one cell made ATP, you pretty much know how the majority of life makes ATP. Yeah. And so like that was really appealing to me. So I sort of went on step by step. I decided to go to graduate school at that time, and the idea of studying single cells was a level at which I could grasp the complexity. I think people end up at different levels, and for me, that cellular level was important. It seemed to me like microbes were a good study system, and so I started there, studied organisms that ate hydrogen sulfide.
So what was your thesis in grad school? What was your dissertation?
Yeah, we're working with this collection of bacteria that made a living by eating hydrogen sulfide.
Okay.
And it's toxic. It's more toxic than cyanide, and yet here are organisms that thrived on it.
Those extremophiles kind of.
Yeah, and so it was like, how does that work? And so my dissertation was focused on those microbes. But I really wanted to do ecology of microbes. Get more towards like a gut system where there are lots of interacting.
Even in grad school.
Yes, but not the gut. I was just thinking of terrestrial, aquatic environments, but the technology wasn't there to do that. Microbiology was studying singled organisms, isolated organisms. So I sort of went along that path. And waited, and fortunately, I remember the day that came out. Norm Pace wrote an article, a commentary. It was in ASM News, 1985, and he said, "Hey, we can just reach into a natural environment, any environment, grab some of the sample, extract the DNA, and we can know who's there." Microbially.
Right.
That was a revolution in microbiology. Didn't have to grow the organisms anymore.
Right. Now, do you mean there's a microbe signature of different people that you can identify with that sample, or are you talking about something?
No, I mean that you can reach in and identify what microbes are there.
Okay, gotcha. Because now I've heard that you can, by sampling the microbes, each of us have a specific signature of microbe that you can identify different people by their microbe content.
Yeah.
It's so crazy.
Dogs too. So there are some apartment complexes that are taking if you're going to have a pet there, you've got to have your dog's microbiome done so that they can track if anybody's not picking up after their dog.
Are you serious?
They know which dog it came from.
That's going on right now?
Yes.
Wow.
Yeah. So anyhow, a little crazy of an application, but right, same idea of what you're saying. And so what Norm Pace was doing was, that's the same technology that we're using now. We target that same gene and use that as
the ribosomal
gene.
RNA?
Yeah, that's it.
That was back in the 80s. That was
1985. That's what Norm said. We can use this to determine what microbes are present in an environment. So for me, that was like all of a sudden you could actually think about looking at the ecology of microbes at that point, like you couldn't do it before.
Okay.
So I went down, did a postdoc with Norm Pace in Bloomington, Indiana, and took off from there. Nowhere near the human microbiome. We were working off the coast of Hawaii in the open ocean. I've spent the last 20 or so years working in terrestrial environments, the microbes in soil that are involved in exchange of greenhouse gases. So it was 5 years ago that I came down to the University of Michigan specifically. It was like, okay, I know how to now look at complex communities, and I thought I had something to offer to study the human gut microbiome.
We'll talk about a complex community. I mean, I was thinking about this the other day, and I was kind of writing it out, how complicated this is. And so I'll lay it out, and you can tell me if there's something I'm missing. So we think now there's 10 to the 14 bacteria. It's 100 trillion, give or take, by power 10.
Huge.
In the gut. Yeah. Most of it's in the colon.
Yes.
Different depending on where you are in the GI tract, even different within a segment of colon. There are different locations even within one segment of colon. So you may have some in the lumen stool, you may have some in the mucus layer along the epithelium, or some attached to the epithelium. They all interact with each other and interact with the gut lining. It's a dynamic system that's changing based on the environment, your diet, all these things. It's different based on your genetic makeup. And on top of that, there's gene transfer going on. So there's genes that are transferring between the different species. So even the identity of a species isn't necessarily straightforward. And then on top of that, you have fungi, archaea, which I'm not even sure what archaea are really, but sort of like bacteria, and viruses that all interact with each other and the bacteria and the human gut. So it's an astounding level of complexity. And when I thought of it that way, I was wondering, where do you even start with that?
Yeah. I mean, you captured it. I mean, it's hugely complicated, and so in some ways we're like where botanists were 100 years ago. We're just trying to identify who's there.
Right.
So what microbes are present? And by the way, when I say what microbes, mostly we're looking at just the bacteria, not the viruses, which is I think hugely important. Mm-hmm. We can talk about the archaea a little bit later. One favorite topic of mine, because I think they play an underappreciated role.
And they're most of the extremophiles, right? That you were probably studying early on with the sulfite.
Yeah, and you know what? Some of them, the ones that we have in our gut, make methane. Mm-hmm. And so you can just take somebody's breath sample and detect if they are exhaling any methane. And if they are, the only place that's coming from or the most likely place is the colon. That the gas that's made in the colon exchanges with the blood, it exchanges in the lungs, and you exhale it. So you can use breath to monitor.
Methane specific for archaea, not bacteria?
That's correct. Yeah. Only archaea make methane. Okay. Yeah. And of course, I mean, hydrogen. Lots of microbes make hydrogen, and you all use that in breath tests
right.
to
bacterial overgrowth. And
yes. But we can return to this too. But I mean, maybe we can start using breath gases to monitor the health of the microbiome as well. Mm-hmm. But anyhow, so where do we start? We start by looking at the composition of that community. Well, what's the relative abundance of things? And like you said, everybody's a little bit different, but there are commonalities. And so I think the first step is to say, okay, well, how's the composition of the microbiome related to obesity? Or how's it related to any condition? And just initially, and lots of the field is still here. We're just looking for correlations. Hey, does the composition, what microbes are there, does that relate to this disease?
Right. And there's a lot of interest in trying to define a core microbiome, right? So if you just take not a disease process, just a population of people lean with no medical conditions, no medication, no antibiotics, is there a certain core group of bacteria that we all share that's necessary for health and well-being?
Yeah.
And that's been very difficult because it's turning out that almost everybody has a unique combination of these bacteria, and there's very little overlap, right?
Yeah,
in the actual bacterial species?
Absolutely. So right. So what species are there seems to vary a lot, but what seems to vary less maybe is what metabolic pathways are there. So maybe there are, a dozen different ways to convert fiber to butyrate. one of the end products that we absolutely need from the gut microbiome. Maybe there are a dozen organisms that can do that, but they all use some of the same pathways. Right. So maybe it's at that pathway level that we'll find some more core functionality.
And that makes sense. If we've all evolved in such different environments, you'd think there'd be some built-in redundancy for resilience of whatever the crucial role they play. It's not necessarily specific to one type of bacteria. There's some overlap there.
Yeah, and like you said, I mean, the genetics and everything are a little bit different, so the host environment might be a little different, and so some microbes might do better in one person than another, but we still need the output from the microbiome, and so I think that's where evolution's acting, is selecting for the function of it as opposed to the composition. So, that'd be one way to explain this paradox that you point out. It's like, well, how everybody's different. So how do you get to some kind of a core? And to me, it's a core function that might give us some insight there.
Right. How much of a problem do you think it is that I think all of the human studies in the microbiome are dealing with stool samples, which don't necessarily address, bacteria that are in the mucus layer or attached to the epithelium. It's almost like a subset, a tiny portion of what's going on in the colon, right? And a lot of the mouse studies, they sacrifice the animals and they study the actual whole colon. Yeah,
it's a challenge. I mean, it's the common approach because it's easiest to get to.
Right.
But you're right, the mucus layer in particular, we've been looking at that, and while lots of the colon has no oxygen, there's oxygen in the mucus layer. We've measured it directly, and so any microbes that are there are going to be influenced by that. And so that mucus-associated microbiome, sure, there's going to be some turnover to it, but it's different than what's in the lumen of the gut, and so it's an imperfect sampling. We hope that it's some kind of summary, that the feces is some kind of summary of what's happened upstream.
Right.
But I think people recognize that it's imperfect.
What do you think the chances are? So there's Helicobacter pylori in the stomach, and that bacteria is responsible for peptic ulcer disease, and in general, the stomach with that harsh environment, there's very little bacteria or other organisms, so it's much easier. What do you think the chances are there's some needle in a haystack in the colon of some direct causation of something down to that specific species level versus I think most of what's going on is going to be a complex interaction, subtle interactions with lots of different things. But you think there's a chance that there is an organism or two that, which is very difficult to find, that may have a more disproportionate influence on health or disease?
I mean, it seems reasonable that there will be some conditions that are like organism specific, and others that are more, a reflection of the community of organisms that are there. But hey, one of the articles I still go back to it that really prompted me to switch over and start studying the gut microbiome was a look at this was a study of mice, germ-free mice versus mice with a conventional microbiome, and they looked in the blood, in the serum, and they just compared what was in the blood of these two groups of animals.
Okay, just for people listening, can you explain just a little bit what germ-free mice is?
Yeah. So mice that are raised without exposure to any microbes. So they don't have bacteria on their fur, in their mouth, in their colon. They're germ-free. There are no bacteria associated with them, and they can be maintained in that. Sort of like the boy in the bell jar.
Okay. So these are mice that don't have any genetic modification. They're just taken and kept sterile from birth.
Correct.
To not get exposed to anything.
That's right.
And you can do that and do experiments in these mice in a practical way and still keep them sterile?
Oh, absolutely. Another reason I came here is because we have a germ-free facility like that. So we're getting ahead of it a little bit, but to test some of the ideas that you had, like, well, maybe one microbe might have a role. So we can monocolonize one of those mice. That is, is just put that one organism in its gut and ask what effect that has.
Gotcha.
So that's one of the biggest tools we have to move forward.
Right.
Germ-free, and so we can put together a single organism, a synthetic community of just a dozen organisms, and that's going to be huge to get to the mechanistic causes that you were talking about.
Right. Okay, so sorry to interrupt you. So you were saying you had the...
Okay, yeah, so there's germ-free and conventionally colonized. There were, if you look in the blood of these mice, in the serum, there were over 300 compounds in the conventional mouse that were not in the germ-free mouse. So, holy cow. I mean, lots of compounds are circulating that are attributable to these gut microbiomes. So if there's one of those compounds that is particularly important in the development of neurological development or disease, then you'd say, oh yeah, one compound, one organism would make sense. But maybe it's a constellation of multiple compounds. So
what kind of compounds were you measuring in the blood?
Oh, I mean, they're mostly small, water-soluble compounds but they range from, fermentation products of the microbes to more complicated or organic compounds.
So you just take a blood sample, do like a mass spec on it or something?
Yes, exactly. Okay. Yeah. Okay. And just say how many elements are in the...
So you might not be able to identify exactly what some of them are, but you can see that by the unique patterns on the spec.
Sure, and so there are labs around the world that are working through that to say, okay, well, what exactly are the compounds and what effect might they have on the host? And, at the other end of the spectrum, well, what microbes are making these?
So, as far as I know, one of the more well-established things that happen in healthy people with the microbes are you have these complex polysaccharides that we eat that we don't have the machinery to break down very well. They pass down into your colon, and the bacteria, so fiber being a classic example, the bacteria break it down into short-chain fatty acids that then get absorbed in our colon and go in our bloodstream. That's the one thing that I think is pretty consistent with most bacteria. And so one mechanism could be those free fatty acids going into the blood, and they're going to cause some upregulation of various different things you could be measuring with that mass spec.
Yes.
But then there could be other byproducts of the bacteria that are getting absorbed as well.
Yep.
So
both things almost certainly are happening, yeah.
And are there any well-known, other than the short-chain fatty acid metabolism and absorption, are there any other well-known things that happen in normal health that are byproducts of bacterial fermentation or digestion that get absorbed into our body?
Something even like serotonin is made by microbes in the gut. It's also made, right? We also make that, but we don't really know how much of it's attributable to the gut microbes and how much to the host. And so that's a pretty complicated molecule. Interacts with neurons in the brain, and so I would give that as an example of kind of a more complex compound that's made.
So, bacteria-derived serotonin gets absorbed into our system.
Yes.
And what percentage do we think? Because I know that about 90% of the serotonin in our body is made from our gut lining. Do we know what percentage of the serotonin is bacteria-derived versus gut-derived?
That might be known, but I don't know. I don't know what it is, and I suspect that it's not. That we don't know that. We know the potential sources, and it's just like,
Do sterile mice get depressed?
It's
a hard research paradigm.
There was one, I was listening to a colleague talk about his studies of germ-free mice, their socialization, and he described them as autistic.
Yeah, I know there's a lot of behavior.
And it's like, wow, that was a powerful statement, and I'm sure he tried to conjure up this image of the potential relationship between brain development and the gut microbiome. But germ-free mice don't behave normally. Their propensity to take a risk is different, and they have people have transplanted a depressive phenotype So mice are coprophagic, right? They eat one another's feces. And so you can put a depressed mouse in with healthy mice and depressed mice. I mean, how do you know that? Right. I mean, there are different tests, like swim tests, like how long will a mouse swim to try to get out of water?
Right.
And the depressive phenotype as well, they don't swim as long.
Or how
long? Yeah, they give up sooner, right? And so there are a number of tests that you can actually measure, and right, we call that depressive phenotype. Right. Are they really depressed? Don't know. But those phenotypes, whether you call them depression or not, you can transplant them by transplanting the gut microbiome. It's crazy.
I've also heard you can transplant a phenotype of obesity. You can take those sterile mice.
Yeah.
And you can take the feces from obese mice and transplant that stool to a group of sterile mice with the same diet. And the half of the sterile mice that get the transplant from the obese mice eating the same caloric intake get higher fat content. And so there are all those really interesting experiments.
Yeah,
like
what's going on there is hard. Hey, and to go back to your comment before, so following that study, people thought, there must be an obese microbiome in people. And so they looked at the composition of the microbes in obese people and lean people. And one study will find one thing, another study will find something different. It's not. So it's, again, something more complicated is going on than just which microbes are there, because there's not been a consistent core microbiome that's lean and one that's obese.
Right.
But, like you say, you can still transplant this phenotype. And so, yes, the microbes are involved, but we don't know how.
It seems like there's this overwhelming feeling that there's something really big going on. And we are just at the very first stages of this. And we're sorting through the noise right now, and it's way more complicated than we ever thought. But I think everybody agrees that there's just so much potential across every aspect of human health and disease imaginable.
I mean, I couldn't agree more, and that's really what drew me. I mean, I was working in a different environments, and this field started to develop, and it was like, wow, I got to get involved in this, and maybe the background that I bring from working with other complex environments will be relevant to this.
Yeah.
So
how much of the excitement you had was with C. diff? Was that a big driver in that? Because that seems to me so far the biggest clinically relevant application of this stuff. And so I'm just going to say real quick what C. diff is. So Clostridium difficile is a colon infection that you are at risk for with antibiotics. So you take antibiotics in some way that causes some dysbiosis or allows the C. diff bacteria to overpopulate the colon and cause this pathogenic situation of C. diff.
Right.
And we can treat it with antibiotics, which has a fairly good success rate. But if you have refractory C. diff, the antibiotics don't work, or you get recurrent C. diff, the best treatment for that is taking a stool transplant from a healthy person and basically recolonizing that colon. And over 90 to 95% success rate on these very difficult cases are cured with these stool transplants. So we had this amazing amount of success with this for C. diff. And then they started finding that there's different rates of asthma and different atopic diseases in children, whether they were born with a vaginal delivery or a C-section delivery.
Yeah.
And they found out that initial, so you're sterile when you're in the womb, and when you come out, that initial colonization, whether you pass through the vaginal canal,
hugely important.
Yeah. And whether it's staph or strep predominant or some of the normal vaginal flora, has implications for all these atopic diseases. So those two things were the things that caught my attention as being so exciting and amazing. So I don't know if you have any other that you know of that really stood out to you as obvious clinical applications of this.
Yeah, you covered a lot of territory there. So the C. diff, that was a good description of what goes on there. So to me, that was when the microbiome got serious. So all of a sudden, this stool transplant taking feces from one person and implanting it to another, and that works better than antibiotics. All of a sudden, the medical community said, we've got to pay attention to this.
It not only worked better than antibiotics, it worked better than almost any intervention for anything.
Yeah.
I mean, the success rate was so high.
That was right. That had a huge impact on the research because it was sort of like what you were implying. It's like, well, if it works for that, maybe it's going to work for other things as well. So I think that really launched the research, and all of a sudden the National Institutes of Health paid attention. Mm-hmm. It's like, oh, so this is actually therapeutic. We're gonna be able to use this, and so to me that was key in launching everything else that we're talking about with the microbiome. Mm-hmm. Yeah, but those. Probably that's the biggest sort of documented success.
Yeah.
So the majority of my time is spent running a research lab, studying something that you brought up before. We're looking at the effect of dietary fiber on the production of short-chain fatty acids in the gut microbiome, and not so much in terms of any particular disease, but just in terms of health. Mm-hmm. Right, so the USDA says that 90% of us don't get enough fiber in our diet. So that to me means we're not taking care of our microbiome, because that fiber is what those complex polysaccharides, that's what's fueling the gut microbiome. So if 90% of us aren't getting enough fiber, we're not, in general, I think, taking care of our microbiomes, and so we're not getting the benefits of the microbiome, and so that's what we're studying, and we're doing it with a cohort of undergraduate students who take a special section of introductory biology, and we offer them the option of participating in a study to supplement their diets with different fibers. And I
was going to ask you, so your fiber is kind of a catch-all term, but there's lots of different types of fiber. So you guys are studying different types and how that affects the...
So resistant starch from potatoes, resistant starch from corn, inulin, which is a polysaccharide from chicory root. So all these are compounds made by plants.
How specific can you get with the pectin and the cellulose and all these different things? Are there foods that really you can hone in on one specific type of fiber, or is there always going to be kind of a mix?
If you were doing the study with foods, it would be more challenging because you'd get all these mixtures. But we're doing it with purified products. So we take resistant starch that's from potatoes, or we take inulin that's been purified from...
So it's like extracts, pure...
Extracts. So it's still complex, but not as complex as just saying, well, eat more broccoli or something like that.
Okay.
So we have these students take a supplement. They stay on their normal diet, so they can eat and drink whatever they do normally. They collect 4 fecal samples sort of before we intervene with...
How do they do that? Practically, how do they have to do that? Because that seems like it would be easy for college kids. They're not going to be too excited to sign up to do this.
You know what? That's what I was concerned about that. I thought, wow, are we going to get anybody that's going to try enroll in this? It turns out that's not a big deal for them. So we have, we can only accept 88 people each semester, and we have over 300 people trying to enroll.
Really? So you're not paying them?
If they complete the study,
they do get a little money.
They get $50.
Okay.
But they have to do a lot of sampling of their gut microbiome. Yeah. That is, they got to sample their feces. They're supplementing their diet and recording this information. So they do a fair bit of work for that, and we give them some compensation for that. But sampling isn't. You want to go into a little bit of detail about how you do it? Yeah. I mean, it's not a big deal. We give them a piece of waxy paper that lays down over the top of the water in the toilet bowl. They produce a sample of their microbiome. Mm-hmm. We give them a sampling kit that has a small scoop on the end. They dip in and get a sample of that. put it right in a tube and screw it closed, shake it up, and that buffer that's in there, the solution that's in there, preserves the DNA from the microbes.
Okay.
So they can they can keep that at room temperature for a day, and they bring it into a collection point on campus, and that's how the study's
easier than I was
conducted.
imagining. I thought they had to be like bent over with some kind of jar. So that's easy. Okay.
Listen, I tested a lot of different ways to collect these samples, and some of them are pretty intimidating. This we arrived at was a very simple, relatively simple way to do it, and it's not a big. I mean, from the feedback that we get from students, it's not a big deal. Mm-hmm. So every semester, we've got 90 people coming in, and it's a new experiment. And so we're able to we're going through these different supplements and saying, how does it affect the microbiome? How does it affect the composition of the microbiome? How does it affect fermentation in the microbiome? And that's one of the cohorts. How did we get here? You said, well, are you overseeing research? So I am, and that's one of the cohorts that we're studying. And we do all kinds of analysis from those. And we can talk about metagenomics and the metabolomics and all that later on, if you like. But that's one of the populations that we're studying. Okay. And it's interventional. We're intervening. We're poking it by giving resistant potato starch and saying, how does it respond? Or how does it respond to inulin? Mm-hmm. By the way, those are common fibers that are added to food. If somebody eats a Fiber 1 bar, that's inulin is the fiber in that one. Mm-hmm. And so we're trying to understand what effect it has on healthy people.
And are you going to extend that to like artificial sweeteners and that kind of thing? I imagine that whatever your assay is can be adapted to any kind of input, dietary input that you want.
Yes. I mean, as you might imagine, the study is overseen by the Institutional Review Board. Mm-hmm. So, I mean, they're making sure that this human subjects research, that we're conducting the research properly without risk with acceptable risk to the participants.
Right.
So, yeah, as far as they'll, as far as we think is reasonable to go with that.
So I do want to hear a little bit more about the process and your method. So you have the sample, and you're having healthy college kids, and you're looking at the one variable you're changing is the amount of this fiber, particular fiber extract.
Yep.
Are they keeping food journals also, or are you just saying so they're keeping food journals? You're kind of keeping an eye on what they're eating.
Yep.
But you're assuming that their diet's not going to change. This is the one dietary change they're going to make. And then you're doing a sequencing analysis of the sample, and you're just basically saying what's there, and you're looking at it at the species level or the phyla level, or what level of specificity do you go to when you're analyzing?
So you're right. We're extracting the DNA from the fecal samples, and we can ask that question at all the levels, at a species level, at a phylum level, and I mean it's an exploration. So typically we're doing it at multiple levels. Where do we see where do we see patterns emerge? In addition to that, we look at fermentation products. So the short chain fatty acids that you mentioned before, they're essential for health. We need butyrate to stay healthy, and we don't make it. Mm-hmm. So we're utterly reliant on the microbes to make butyrate, and there are health consequences if we don't have enough butyrate. Inflammation is associated with that. Colon cancer is related to the amount of butyrate that's produced. So there are real consequences of not having a microbiome that's making enough of that particular short-chain fatty acid. So we measure those. We measure the DNA. And yes, they do the dietary logs. And by the way, that's their end of the semester project. So in that week before they start any supplement, that's when they do the dietary logs. And so we're asking the students to help us figure out how diet is related to microbiome. Mm-hmm. So we've had hundreds of students now go through, and we can look at vegetarians and vegans and the amount of dairy consumption and ask whether or not there are any relationships with those aspects of their diet and who's there, what microbes are there.
But how do you know what to look for? So you have all these stool samples from all these college kids. Are you doing a full sequence analysis of every stool sample?
Yeah, we are.
And
so each semester, that's 8 samples times 90, so that's 720 samples that come in.
Okay.
And I mean, we've got the setup, a robotic setup to extract nucleic acids, and we can get DNA sequence from all of those. And so
down to what level? So if one of your students gives you a sample and you sequence it, can you tell them exactly how many species of each bacteria they have in their colon, or can you tell them you have this percentage of firmicutes and this percentage of Bacteroides? What level of...
Yeah, well, we get a pretty... I mean, we sequence pretty deeply. We get a lot of sequences back, but it's always in terms of relative abundance, like you say, a percentage. So it's not an actual number of microbes.
Right.
It's what's the composition of
delta,
that.
the change.
Yes, or just percentage. So while you're during that first week of the study, before you started any intervention, what's your microbiome look like on average? Okay. So we take 4 samples and say, okay, average those 4 and say, okay, here's what your microbiome looked like during that week, and here's what your average diet looked like during that week.
And that's to the species level, not the phyla level.
Yeah, right.
Because when I read about stuff, it seems like a lot of times they're talking about the phyla level, which seems like it's not specific enough to really...
we go very... we do both. And I think what people are doing is they're just looking for, at this stage, they're just looking for patterns.
Right.
So if the firmicutes, which is a big grouping of bacteria, if that shows some relationship to obesity or something else, then that's what's reported.
Right.
And that usually means that they didn't find a relationship at a more specific level. And so you sort of report wherever the patterns are.
Yeah.
And again, that's a stage where a lot of this is just finding patterns, and then the challenge is to figure out what's going on. Yeah. Why is there this pattern?
Yeah. So, have you seen any patterns yet? Any big outliers of something consistent that happens with the fiber in these?
Yes. Glad you asked. So about 2/3 of the people who consume these different supplements, and the one that has had the biggest effect so far, I can tell you, is resistant potato starch. About 2/3 of people, their microbiomes increase in butyrate production. And we have a good idea now of what configuration of microbes is responsible for that. And so what we're doing now is isolating those organisms, and we'll go back and put those together in the germ-free mice that we were talking about to test that hypothesis. But if I can, let me take this in a little bit of a different direction.
Yeah.
Based on that result, we're in a clinical study. So can we go there now? So lots of people who are treated for leukemia ruins their immune system, their bone marrow, and so they come in for a bone marrow transplant.
Right.
So that bone marrow transplant is a graft from another person, matched as best as you can match them, and in a number of individuals, that graft rejects the host.
Right.
So this is the graft versus host disease.
Yeah.
So a colleague of mine here at Michigan showed in a mouse model that if he applied butyrate to the colon, he could decrease the incidence and severity of graft versus host disease.
How's that possible?
Oh my goodness! So, all right. So we'll come back to that in just a minute. But he published that in 2016, and we published about the same time. We published that we could increase butyrate production with resistant potato starch. So we are currently in a clinical trial. People who are coming into the University of Michigan for a bone marrow transplant, if they elect to, they can enroll in this study, and they're receiving resistant potato starch as a supplement to their diet, because, we're hoping to prevent the development of graft versus host disease.
By increasing the butyrate.
Exactly. So we're assuming a couple of things. One, that in this group of people, we can have the same effect as we do with a healthy cohort of undergrads, and that the people will respond the same way that mice respond. So lots of assumptions, but it was a strong enough indication. It's like, well, I mean, it would be irresponsible not to try this. Yeah.
Is the rate, or is the incidence of graft versus host disease higher in mice that are sterile? Do we have any data on mice that don't that are sterile guts that get transplanted?
Yes. Yep. And so if you transplant in organisms that can make butyrate, you can decrease that incidence of graft versus host disease in mice. Or you don't even need the organisms. You can just put butyrate in.
What if you just have a sterile mouse? So you have a mouse that does not have any gut biome, and you do a bone marrow transplantation in those mice versus wild-type mice.
Yeah.
Is the rate of graft versus host disease higher in those?
So it gets difficult technically to remove the mice from that sterile environment and do the surgery and keep them sterile. So we can't.
Well, you would predict that it would be
yes,
much higher
absolutely. And if you apply butyrate, and so Pavan Reddy was who's head of the transplant center, did this research in his lab, and he showed that butyrate was affecting transcription of genes in the host cells that line the colon. So he has a reasonable mechanistic explanation for how that's possible.
Okay,
so
say that again. Sorry, I missed
it. Yeah. So he applies butyrate, and he sees the expression of genes in the host cells lining the colon changes. And it changes in a fashion, and there's a whole cascade of events that's triggered by butyrate. So he has a reasonable explanation. So you reacted like, how's that possible? We can walk through that on a butyrate affecting gene expression. That and a whole cascade of events that follow from that. So there's a plausible. In that case, it's one of the few cases where there's really a plausible mechanistic explanation from gut microbe to metabolite to host gene expression. So I'm delighted that we're taking results from this healthy cohort and applying it to a clinical cohort. I don't think it's going to work because
most things don't. Welcome to the science.
Well, that and people that are coming in for bone marrow transplants, they're just getting whacked with antibiotics prophylactically. Yeah. So it's almost like they're handing us a depleted microbiome, one that's been hit with antibiotics, and saying, here, make butyrate. Mm-hmm. And it's just like, I mean, it'd be great if it works, but in addition to a lot of things not working, that would be one reason that it wouldn't work. But again, if we can figure out what the organisms are that we need, maybe in the next iteration we give not just the resistant starch, but we give the organisms that are necessary to convert that to butyrate. Right. So, there's a way forward. Mm-hmm.
So that's interesting because I've known about butyrate a little bit because there are some applications of inflammatory bowel disease where you give butyrate enemas and it's been shown to be beneficial. So that's one mechanism. But another thing that I've heard.
By the way, I understand that nurses hate when you order a butyrate enema.
Yeah, they're not big fans of any kind of enemas.
But that's it smells bad.
The butyrate itself smells
acid.
Bad? Oh yeah, I didn't know that.
Oh yeah, no, it's.
I think nurses are.
It's a putrid. It's a putrid
they're like
smell.
immune to smells at this point. You got a tough job.
Yeah.
So another thing I've heard is like when you said, we're kind of step back and just at least see patterns to start with. And one of the patterns they seem to find is that the diversity of the microbiome is important. And in a lot of these disease states, when they analyze the microbiome, the one thing they're finding, maybe there's no consistent pattern other than there seems to be a less diverse population microbiome. Yeah. And so butyrate production is kind of a non-specific metabolic pathway of most of the bacteria. You would think that if if you eliminated the diversity by 50%, as long as you're producing the butyrate, which I was under the assumption that they all did, then you'd still be okay. So obviously there's probably multiple mechanisms going on. But do we know that different bacteria produce different levels of efficiency of producing the butyrate with a given?
Oh, absolutely.
So
there's
across the board of.
No. That's a specialized fermentation product. Mm-hmm. So I could name 6 genera of bacteria that are the most likely those that are producing butyrate.
Okay.
And like you said before, there are hundreds of different microbes there, and so we need that. But I always go back. People ask if butyrate's so important, why don't just give butyrate to people?
Right.
But I don't think that. I don't think that's the whole story. I go back to this 300 compounds circulating in the blood,
right.
And it's like, okay, well, butyrate's one, and yes, maybe that's the key for graft versus host disease, but it's not the key for health in general.
Right.
And so, we zoom in on some specific questions and metabolites, but I always kind of zoom back out and say, hey, listen, that's only one of many compounds that are produced.
But do we know just to stick with the butyrate idea, do we know all of the different species and their capability of producing butyrate? So if you looked at, did a meta-analysis, a bunch of studies that showed you couldn't find any other patterns other than patients with ulcerative colitis had a decreased diversity of the microbiome, and you analyzed to see if the amount of bacteria that were eliminated were the higher butyrate producers, and the ones that were left behind were the low butyrate producers, that kind of thing.
Yeah.
Is that something you would theoretically be able to do?
Yeah, absolutely. And so it would probably not be looking at, in that case, which species are there, but it would be taking all the DNA and sequencing all of it so that you could actually look at for that pathway that leads to butyrate. So you could say, regardless of what organism holds that pathway, you'd zoom in on the pathway and say, okay, well, how abundant is that pathway? And again, like we were talking about before, maybe there are a half dozen different ways to put together that butyrate pathway. But that's a tractable problem. That could be answered. Yeah.
Are there any insights from identical twin studies of trying to get at how much the genetics of the host influences the microbiome?
Yeah. So there have been studies both with identical twins and just more general studies trying to link the human genome with the gut microbiome. And they're underway. I'm not aware of any clean answers that have come out of that. And for those GWAS studies, for those genome-wide analysis studies, you need thousands and thousands of people. So I think we're still gathering data, and it's going to be harder. Right, it's hard enough to do that with some diseases, genetic diseases, and so we've got, like you mentioned, diet is affecting it, and so it's a challenge,
And I'm curious. So I'm surprised that you're able to do that level of resolution for your genetic analysis. Like, it's gotten that cheap. So it sounds like cost, in a big way, is less of a barrier for doing these kind of analyses.
Agreed. The barrier now, the bottleneck now is analyzing the data, and analyzing the data with some ideas and with some hypotheses. And so I sit on a number of advisory boards for other microbiome centers, and they're usually very strong with the clinical aspects. They're very strong with the data gathering and modeling, but oftentimes they don't have microbiologists there to say, well, this is how you put these things together. So we're definitely stuck, or that's the bottleneck now. It's not so much collecting the samples. That works pretty well. It's not so much extracting the DNA or generating the data, generating the sequence data. It's like, okay, here you go. Here's 3 terabytes of data. What do you make of that?
Yeah. What do you think about, what surprises me is how much diversity there seems to be from person to person with the signature of microbiome that they have, right? So you take 10 healthy college kids, and there's surprisingly less overlap maybe than you'd expect in their microbiome signature. And there's also fairly little variation over time.
Yeah.
In the microbiome. So, they were so different. So you think, okay, it's just a really dynamic process. It changes every day, and so you take a snapshot at any given time. That's why they explain the differences. But then you take an individual person, and you do that analysis, and you follow them for a month. It changes very little, right?
Your microbiome always looks most like your microbiome.
Right.
Yeah.
Unless you take antibiotics, and then even then it comes back. It usually gets back to what you were, but it's remarkably stable, even though there are so many different environmental factors. So diet, the air you breathe, right? There's learning that there's 10 to the 5th power bacteria floating around, viruses. How often you brush your teeth. The girlfriend you're kissing, the traveling that you do. All of these exposing you to a huge difference in.
And it perturbs it for the short term, but then it comes back.
Right back to where it was.
Even with these studies that we're doing with the different fibers of resistant starch, we while people are consuming that, it changes their microbiome dramatically. But when they stop, kind of goes back to where it was before.
Yeah.
So
I wonder if that's the missing piece. I wonder if it's the resilience that's missing in disease. That the people that get disease from the alterations in their microbiome
knocks them into some other.
They're just not able to.
Yeah, that's
whatever
interesting.
The process is that
I like
brings
that.
people back.
Yeah. And like you say, typically with antibiotics, people bounce back, or mostly so anyhow.
Right.
But how far can you push that? At some point, that ecosystem breaks down, and it goes into an altered state. Probably that's happening in the recurrent C. diff that we were talking about.
Yeah.
It's just too far away. It's not going to recover. And that's where stool fecal transplants might come back in. I'll tell you what. If I'm admitted to the hospital, I'm going to save a sample of my feces in my freezer at home in case. In case it gets pushed too far out of balance, I want to be able to restore it.
Yeah.
So
that's another interesting. Before I get into that, the people that get C. diff are old people.
More commonly,
you get antibiotics throughout life, and occasional young person will get C. diff, but most people can bounce back. It gets back to that resilience, and something happens when you're an old person, you get antibiotics, we get C. diff, whereas you get antibiotics when you're a young person, your body somehow finds a way to recuperate, be resilient, get back. So it's back to that issue of, but what you're mentioning about saving your own stool, my last podcast was about stem cells, and I had a bone marrow aspiration, injected it, I got a bum shoulder, and I stored some of my stem cells, because there's a lot of research on stem cells of systemic therapy with stem cells and whatever. So now I have a bank of my own stem cells when I'm 80 if I need them. Which reminded me of when you said you're going to stay.
Well, so you might stay.
So I should
put
add
next to that.
Add something else in the freezer. But the stool transplants so far have been done mostly with other people's stool. So sometimes family members, sometimes not.
Right.
They do some rudimentary tests to make sure there's no infectious disease going on or anything like that.
Yeah.
And there's been very low complication rate, very little, adverse events with that.
I mean, at least in the short term.
Right.
Yeah.
But it's interesting to think of, these phenotypes you see when you do these stool transplant in the mice. Like, are there things you can transplant into people based on the microflora from other people? And I joked with somebody when I was a fellow like 5 years ago that there's going to be people selling like Lance Armstrong stool or some model stool, or you get a transplant with some model of someone who's thin, and you want to, and there's these signatures of these different people, and and I was joking, but now there's people looking into this as far as obesity and disease and health. Is there something about their microbiome that's contributing to their health? And I want that transplant into me. So it's kind of a bizarre detour on this stuff.
There are just a couple of studies out about these methane-producing archaea in the gut. And the one thing that's kind of interesting is that endurance athletes seem to have more commonly have methanogens in their gut microbiome than the rest of the population. Huh. So, like you're saying, Lance Armstrong, I mean, maybe, we want that high methanogen community.
Yeah.
But, right, you probably have to change something else so that it's preserved, so that it keeps going, because
or else it'd be like any other alteration.
It bounces back
later back to where you were before.
Right. So probably it'd be great if that was the case, that you could just transplant it and get all the good things associated with it, but probably wouldn't work.
Yeah. So for you in the future, what are the most exciting things that you see going forward? Do you think it's just a matter, like you said, of just getting through that bottleneck of all this data and analyzing it, or do you think the field is kind of being held back also by some kind of technology that you need to break through, or do you think it's just a matter of just going through all this data?
Combination of it. One of the things that you mentioned earlier is the structure of the microbiome, next to the mucus or in different parts of the GI tract. And we destroy all that, right? We just take a sample and grind it up. So we don't preserve any of the three-dimensional structure of it. So in particular, you mentioned like associated with the mucus. It's like, hell, we don't know what's associated or not. We just grind it all up. And so I think that's one of the limitations that we have right now. We don't know what the architecture of those microbial cities are. We grind it all up and then look at it together. So I'd say technically that's one limitation that we have.
Have there been any comparative studies with some type of result in a mouse where they compare taking a stool sample from the mouse and taking a colon sample, sacrificed mouse, and sequence that segment of colon versus the stool before they sacrifice, and see if there's some concordance?
It's different. There's some biogeography. Mm-hmm. The mucus is different from what's in the lumen.
Right.
No
but
question.
You see similar changes. So I know there's different compositions. Let's say you find in this particular phenotype you have a higher percentage of firmicutes in the stool. Have they ever compared? Okay, we also see a higher percentage of firmicutes from the biopsy versus the stool.
Oh, I see. You're suggesting developing a framework so that we can use the stool samples.
Yeah,
and get a little better. Yeah. So we know it's. we know it's not perfect, but I don't think we have a very good way of using that to say, okay, well, you're underestimating this by so much, or in this case, you really need to go in and do a biopsy to get a sample. Yeah, we're not that far. So so there are some technological limitations. Ultimately. Maybe we don't really care so much about what microbes are there or what genes are there. It's really the products that we're interested in, and so that's a limitation. Mm-hmm. We can get to the to the DNA and to the RNA more easily, but what the metabolites are, that's a little harder to get to.
Yeah.
You got to preserve the samples a little bit differently. They can't, and so technically there's that's what we really care about, right? How are the microbes communicating with us?
Right.
And it's not through their, it's through the products that they make. And so we're probably underpowered there in being able to do that.
But at least at the level of sequencing, you think that's not holding you back. So it's a rigorous, repeatable, reliable. So, for example, if you took genetically identical mice and you took them and you raised them in the same cage with the same food supply, and you analyzed their microbiome multiple days, say 3 days in a row, you would imagine there would be almost completely overlapping in the microbiome, and it wouldn't change from day to day, or at least it would change the same way from day to day.
That's generally true. So I think it's not so chaotic that we can't make sense out of it. So we're at the stage where technically we're tracking a community of microbes, and I think we can do that with a reasonable level of confidence. Okay, good, we don't get any of this geography of the gut and that kind of thing, but just taking fecal samples day to day, same mice, given the same diet, that works pretty well. Of course, if you do that experiment and then do the same thing, same diet and everything, but in another location, like at another laboratory,
right?
Or now you get some changes, and unclear why. It's like, hey, well, the food comes from the same vendor. Everything is the same as far as we can tell, and we're trying to repeat studies at different institutions, and well, there are differences. Yeah. So,
have you analyzed your microbiome?
Sure have.
And there's a public database where you can do that, right?
Yes. Yeah. We just do it in my. I just do it in my lab.
Yeah, you can do it on your own.
Yeah, right.
But
right.
I think there's a resource where people can go online and get a full readout of their own microbiome.
Yeah, the American Gut is one of those. So they send you kits not dissimilar from what we're using to collect a fecal sample, mail it in, and they'll extract the DNA and tell you what's there. And so. I mean, I think people do that out of curiosity. We're probably not yet at the stage where you can learn too much about, like, well, I don't know, if we handed you an analysis of the microbiome, I mean, how much could you diagnose about the gut health from that? I mean, that's pretty hard to do. So maybe it's a little more of a curiosity thing at this point.
And maybe just adding to the database of information data out there, but
well, that's huge, right? I mean, it just gives a bigger pool. Yeah, what's the American gut look like?
Right. Yeah. There's a lot of interesting implications too with factory farming and antibiotics for livestock, and how much of that is contributing to changing of our gut flora over time, and is that related to this spike in obesity that we seem to be seeing, this epidemic of obesity that we're seeing? And there's just so many other interesting ways we can see it going.
Yes, and the microbes can change quickly, like you say. I mean, they pass DNA amongst themselves. Antibiotic resistance is a good example. But, I mean, we've seen, maybe you saw this study where the capacity to degrade some of the compounds in seaweed was found more commonly in a population that was, I think it was a Japanese population that was eating more seaweed and sushi. Yeah. So it's like, okay, well, that's a relatively recent dietary thing, and the microbes have responded. Picked up a gene to do that from marine bacterium.
Right.
It appears. Yeah.
Well, Tom, you've been so cool to do this, generous with your time. I really appreciate it. Anything else you want to say about your future plans in the lab or anything?
Oh my goodness!
Recruiting any grad students out there?
Yeah, we're not yet there, but I'd like to get to the stage where we can involve the community more. I think people are interested in this, and I would like to be able to expand. We don't have the mechanism in place of the funding, but I'd like to expand to the point where the community gets involved, in particular the elderly. Like you were saying, that seems to be a population that is more susceptible to C. diff infections, and my suspicion is that we need to pay more attention there to the gut microbiome. I think it's less resilient, and while you're in college, it seems like you can bounce back from a lot of things, and your gut microbiome can bounce back from a lot of things, but maybe that's not...
microbiome has anything to do with hangovers and drinking, because I can't tolerate like 4 beers anymore. I wonder if a stool transplant I can drink like I was in college.
Well, that might be a harder study to approve by the IRB,
Right,
but you know.
But it's got to be exciting because, like you were saying, I mean, it's almost unlimited potential of, at least at this point, you can see how it connects with brain science.
Yeah.
Almost any mechanism you can imagine, it's going to interact with your gut lining, with your immune system, with the microbe population there. And
yeah, it's an exciting time. I just hope that we can deliver on some more things like the stool transplant and C. diff.
Yeah.
Because at some point, people are going to get tired of the hype.
Yeah.
And say, we already heard all this. You guys said that the microbiome was related to autism, and it's related to this, and it's related to that, and we don't see any result.
Yeah.
So I don't know how long it's going to last.
Yeah.
So we got to get cracking.
It's true, you know.
And say, okay, well, look, there is a relationship here, but there's not here. And so, I mean, we've got to get beyond the correlations that we're at now. We got to nail down some of these. And so if we can have a success with graft versus host disease, or if somebody can have an effect with metabolic syndrome and obesity, I mean, that would be huge. But they're going to be, some of these correlations are going to be just coincidental.
Yeah.
And so that's what we got to do with it, man. We got to figure that out.
Well, the C. diff thing seemed to pop out of nowhere. I mean, this is before there was any serious money being put into this kind of research.
That's right.
It was very recently that all these resources have been put into this field. And so I think that it's just a matter of time. And there's always going to be a lag.
Yeah.
All the hard work that gets done in the bench, on the bench side of things before it turns into something clinically relevant. But
yeah,
I think it's going to happen.
Yeah.
For sure.
I hope so. I hope so. But right, I mean, it's like, yes, the NIH is pouring a lot of money into this, and at some point it's going to be like, all right, well, where has that investment? We've been investing a lot of taxpayer money into this, so where's that investment got us?
What has he done for me lately?
And so, yeah, no kidding. I mean, yeah.
So my last question for you is, do you think, I've been wondering about this, me and some of the GI fellows are talking about it, are we better off or worse off in the hospital dealing with all these C. diff patients? So I'm definitely probably colonized with C. diff. Is that in the long term going to be a positive thing with my immune system adjusting to that, or am I just set up to be at more risk for C. diff when I'm older? And I was wondering, it'd be an interesting study to take people in the healthcare field that retired and they've been working for the last 20 years, say. They've retired within the last 5 years.
Yeah.
Just do a retrospective study and see how often they've been diagnosed with C. diff compared to the general population. And I wonder if it's higher or lower.
Yeah. What an interesting question.
Know.
Hey, I mean, just to do that survey now to say, hey, in fact, just because you're in the healthcare system, is carriage of C. diff more likely?
Yeah.
I mean, I don't think we know the answer to that, do we? So just to see if it's higher, but then you had to do it prospectively. So that'd be the other thing that we probably need. It's sort of like the Framingham heart study where they look,
Framingham stool study. That's what we need.
How much does your microbiome now affect what's going to happen when, 20 years down the road?
Right.
And same, for these stool transplants and the antibiotic use and everything else. So, yeah. But I agree, man, exciting times. And, I hope that we can come back down here in a couple of years and say, all right, what do you got for me?
Yeah.
So what's the last 2 years given us?
Absolutely. All right, man. Well, I really appreciate it.
Great. All right. Cool.
Great talking to you.
Likewise.
So that was a good one. Hope that was interesting. I'm definitely going to follow Tom and see where his research goes. I totally failed to come up with a song for this episode that matches the theme. So this is just a random song I like by Neutral Milk Hotel. And this might be my last podcast. Not sure. Nothing planned in the future, but you never know. So thanks for listening to any of these if you have, and maybe there'll be another one sometime, maybe not sure. All right, see you later.